Hello students, so this is the lecture for 10th class SSC board, subject science and technology part 2. So we are going to start with our first lesson, that is heredity and evolution. So first, heredity. So we all have heard about this word heredity, and we all know the concept of the heredity. So that is nothing but transfer of characteristics from parents to offsprings, means from one generation to next generation. So transfer of characteristics. And the study of the heredity is useful for diagnosis, treatment, and prevention of hereditary disorders, production of hybrid varieties of animals, plants, and in industrial processes in which microbes are used. So there is a very vast use of the study of heredity or science of heredity. Here just 2 or 3 points are mentioned in our book which are related to us, which are related to our study. So hereditary disorders and the industrial processes are the applications, or they are the useful study is useful for mainly these areas. So as we are studying heredity, this is the very important point related to that, that is DNA. Let us see how. So first DNA, let's divide DNA, which we have studied in 9th standard. Its full form is deoxyribonucleic acid. So where do DNA exist? So it exists in cell of each and every living organism. So look here, this is the cell. So it is present, DNA is present in the nucleus of the cell, right? Are you recalling it? It is present in the nucleus. So chromosomes are present in the nucleus and chromosomes are made up of DNA. So diagram simply shows that a cell, its nucleus, in the nucleus chromosomes are present. So these are the chromosomes. So this this is the chromosome, which is made up of, it shows that it is made up of the double helix structure, that is DNA, right? And one part of DNA is called as gene. So genetics, you have heard this word, so heredity, genetics. So gene is also important point. Okay. So we should know at this stage, we just should know that where does this gene, what is the gene, what is DNA. So gene is some part of DNA which is present in chromosomes, which is present in nucleus of the cell. So what is RNA now? So we have studied in the 9th standard that DNA is double-stranded and RNA is single-stranded. But RNA is nothing but a disposable copy of segment of DNA. So it is coming from DNA itself. It is just a copy of DNA which is disposable. We will see how it is disposable. And types of the RNA which we have already studied in the 9th standard, that are mRNA, that is messenger RNA, rRNA, ribosomal RNA, tRNA, transfer RNA. We have studied the types of RNA and their functions in one line in 9th standard. Here we are going to apply the functions in the process next explained. So this is the process that which we are going to study. So it is the first point which is covered in this first video, that is protein synthesis. So protein synthesis is nothing but production of proteins in the cell. So the process, this is the process all cells use to make proteins which are responsible for all cell structure and function. The cell itself produces proteins in it which controls the function of the cell. So each cell produces protein, and DNA in the nucleus of the cell carries the information necessary for this process, that is making of proteins, that is protein synthesis. Different cells, different types of cell produce different types of protein. For example, you have heard about this. The blood cells are one type of cells, right? So blood cells produce hemoglobin. Skin cells, they have melanin. So we are going to study in detail in the next lesson different types of protein produced by different types of cell. But here we are just taking the examples of simple. Blood cells produce hemoglobin. So different cells, according to their function, produce different types of proteins. Now the steps of protein synthesis, which are important steps, those are transcription, translation and translocation. So don't get confused, because we are going to study the 3 steps in detail now. Okay, so the protein synthesis is nothing but protein making, production of protein inside the cell. So each cell makes protein, and the process in the cell, that is protein synthesis, that we are going to study now. Okay. The first process is transcription. So what is transcription? So look at the diagram. So we can see here, look here, in blue color the DNA is shown, right? And one part of it is gene, and from that DNA the red color strip is shown, right? So from the DNA in this diagram, okay, so the colors are not like this actually in real. So this red is named as this, we can see this red strip is known as messenger RNA. So DNA is transformed into RNA. Let us see. So DNA has the genetic code for the protein that needs to be made. We have seen in the previous slide that DNA has the information, right, for protein synthesis. So DNA will control the process of protein synthesis. It has the information which is needed for protein making. So it has the information, but it cannot go outside the nucleus. It is in the nucleus. Look, this is cytoplasm of the cell. So whole, this is the cell. Then this violet color, purple color is this one is part of nucleus where DNA is there, right? So DNA cannot come outside the cell. Therefore, it transforms that genetic code, genetic information for protein making into messenger RNA. So the messenger RNA, it is transformed into messenger RNA. So DNA has the, read here, DNA has the genetic code for protein that needs to be made, but proteins are made by the ribosomes. But the proteins in making process is performed in the ribosome, which is outside the nucleus, right? So therefore it needs, that information needs to come out, so it can be come out through the disposable copy of DNA, that is nothing but RNA. Okay, so transcription is nothing but DNA to mRNA, that is messenger RNA, to come out that information, for coming out that information from nucleus to cytoplasm. Okay, so you must know the structure of the cell. There is nucleus in the center, and outside that there is cytoplasm and other cell organelles. So the genetic information in the form of RNA comes out from the nucleus, from DNA, in the form of RNA towards cytoplasm, where the ribosomes are there, right, in the cytoplasm. Now the sequence of nucleotides, you must know what are the nucleotides. In the DNA or RNA, they are made up of nucleotides, sequences of nucleotides. The nucleotides are adenine, guanine, cytosine, thymine. And in RNA, the same nucleotides are there, adenine, guanine, cytosine, and thymine is replaced by uracil, right? Now, RNA is nothing but the disposable copy of DNA. So the sequence on the nucleotides of the RNA, look here in this diagram, first DNA is shown, right? Then some part of DNA is shown, it is zoomed in, right? Then from that, so this is the part of DNA, this is the part of DNA, and in green color mRNA is shown. So how mRNA is formed? It is complementary, the sequence is complementary to DNA. Look here, first A, for A, the pair of the nucleotide is uracil, adenine, for adenine in the RNA, that is uracil. Then for thymine it is A, for guanine it is C, for cytosine it is G, means guanine, for thymine it is A, A means adenine. If there is adenine on DNA, there will be uracil on RNA. So this is nothing but complementary sequence. So the sequence will be complementary to the DNA strand used for the synthesis. Genetic information from the DNA is complementary to the the genetic information transferred by mRNA, messenger RNA, is complementary to the sequence of on nucleotides on DNA. Getting it? So look at the process through the animation. Start. So this is the nucleus from where mRNA comes and go into the ribosome. This is nothing but transcription, simple transcription. So transcription means genetic information present in the DNA which is necessary for protein synthesis, protein making, comes out of the nucleus to the cytoplasm through RNA, that is mRNA, messenger RNA. Okay. So look at it again, and enters into ribosome. Okay. Now the next concept, triplet codon. So we have seen transcription. We are going to see the translation. Before that, you should know the concept triplet codon. So we have seen the messenger RNA, mRNA, right? So each nucleotides, three nucleotides in the messenger RNA are called as codon. But what is codon? So look at the definition. So each message on the messenger RNA transforms, passes the message, right? So each message, that coded message contains the codes for amino acids. So this message or this nucleotide sequence of nucleotides are nothing but sequence for codes for, it contains codes for amino acids. So you know that this is mRNA and the sequences are like this, right? But this message contains codes for amino acids. So code for each amino acid consists of three nucleotides. So three nucleotides forms a code for one amino acid, that is called as codon. So codon means three nucleotides on mRNA, simple it is, and they carry information for code for one amino acid. Okay. Now tRNA is also being used, okay, in the process. So each tRNA has three, this is the tRNA, okay. So each tRNA has three nucleotides that are complementary to the codon. Okay, so this is the codon, and tRNA has anticodon. Okay, mRNA has codon. You will get the process and all the things in the next slide, next 2-3 slides properly. Just remember right now that codon is nothing but three nucleotides which carries code for amino acid on the messenger RNA, that is called as codon. Okay, and tRNA has anticodon which is complementary. Okay. For example, if it is G, tRNA will have C. If it is C, tRNA will have G, because that is pair. Okay. If it is A, tRNA will have U, uracil. It doesn't have thymine nucleotide. Okay. So it has complementary sequence to the codon. Okay. That is called as anticodon. Okay. Here these are not same. This anticodon is not for the codon. Okay. So next, so you have we have studied the concepts, what is codon, what is anticodon. Codon on mRNA and anticodon on tRNA, which is complementary, which make pair. Okay. So now the second process, that is translation. Translation means nothing but making protein, actual production of protein. Look at the diagram here now. Now you can see this is the mRNA. Can you see this? This is mRNA. So the whole strip is mRNA. Now as per the sequence on mRNA, as per the sequence on messenger RNA, Look here also, the names are given mRNA. And the process is performed in ribosome. Okay, the process is performed in ribosome. So therefore this is the whole, this is blue and this yellow is ribosome, that is one cell organelle. Okay, and according to the sequence on mRNA, according to the codons, means three nucleotides, group of three nucleotides. According to the codons, the tRNA are attached to the codons, to the mRNA. Look at this. First, this is look at this. This is translation direction. So first, for AAA, tRNA has anticodon. So UUU. For UUC, it is for UA. A for C, it is G. So it is attached, right? So according to when the codon anticodon are matched, so according to that code, means code is transferred, according to that code, amino acids are brought up there, and then all the amino acids are brought up there, and all the amino acids brought up are linked with the peptide bonds. They are linked. So each tRNA, when it is attached, when they are matched, the code is matched, the amino acid is brought there in the ribosome, and the amino acids brought are attached together by bonded together by peptide bonds. Here you can see. Okay, so now let us see the translation. So translation we use for translation. Translation म्हणजे काय तर, for example, translation English to Marathi translation. सो त्या भाषेत ज्याला जी भाषा कळत नसेल, उदाहरणार्थ कोणाला इंग्लिश कळत नसेल, तर ते आपण ट्रान्सलेट करून सांगतो, मराठीत सांगतो, राईट. सो हे ट्रान्सलेशन झालं, त्याला कळेल अशा भाषेत. सो कोडॉनला जी भाषा कळते अँड ट्रान्सफर आरएनए ते ती भाषा त्यांना कळते आणि तशा पद्धतीने ते त्या पद्धतीने डिकोडिंग करून ते अमिनो ऍसिड घेऊन येतात. ओके, मी एक एक्झाम्पल पण सांगेल एंडला. सो आतापर्यंत मी मराठीत बोलले नव्हते, एकदा मराठीमध्ये सगळी प्रोसेस सांगते स्टार्टिंग पासून. आधी आपण बघितलं डीएनए, डीऑक्झी न्यूक्लिक ऍसिड, डबल हेलिक्स स्ट्रक्चर ज्यामध्ये न्यूक्लिओटाइड्स असतात, ॲडेनाइन, गुआनाइन, सायटोसाइन, थायमाइन, राईट. देन आरएनए दॅट इज सिंगल स्ट्रँडेड, त्याचीच कॉपी असते, ती फक्त सिंगल स्ट्रँडेड असते, त्यातला एक सिंगल स्ट्रँड म्हणजे आरएनए. त्याच्यामधले न्यूक्लिओटाइड्स वेगळे असतात, ॲडेनाइन, गुआनाइन, सायटोसाइन अँड थायमाइनच्या ऐवजी युरॅसिल. ओके, सो पहिली प्रोसेस प्रोटीन सिंथेसिस म्हणजे काय तर प्रोटीन प्रोडक्शन ऑफ प्रोटीन. प्रत्येक सेल, बॉडीमधली प्रत्येक सेल ही प्रोटीन्स बनवत असते, का तर त्या सेलच्या फंक्शनसाठी नेसेसरी प्रोटीन्स बनवत असते, त्याच्यावर त्याचे बेस्ड. फॉर एक्झाम्पल ब्लड सेल्स हे हिमोग्लोबिन बनवतात, फॉर्म करतात. प्रत्येक सेलचं काम असतं प्रोटीन बनवणं स्वतःला लागेल असं, स्वतःच्या ग्रोथसाठी, स्वतःच्या फंक्शनिंगसाठी. ओके, सो ते जे प्रोटीन्स आहेत ते बनवण्याचं काम म्हणजे दॅट इज प्रत्येक सेल करते, ती प्रत्येक लिव्हिंग सेल करते, दॅट इज नथिंग बट प्रोटीन सिंथेसिस. ती प्रोसेस आपण बघतोय, त्यातल्या 3 स्टेप्स बघतोय आपण. काय तर ट्रान्सक्रिप्शन, ट्रान्सलेशन, ट्रान्सलोकेशन. त्यापैकी ट्रान्सक्रिप्शन बघितलं आपण. ट्रान्सक्रिप्शन म्हणजे काय तर डीएनए न्यूक्लियस मध्ये असतो. प्रोटीन सिंथेसिस न्यूक्लियसच्या बाहेर सायटोप्लाझम मध्ये होतो. सो प्रोटीन सिंथेसिस साठी नेसेसरी असणारी इन्फॉर्मेशन ही मात्र डीएनए कडे असते. जे काय आहे ते डीएनए इन्फॉर्मेशन पाठवणार आहे, त्याच्यानुसार प्रोटीन्स बनणार आहे. सो डीएनए ची इन्फॉर्मेशन सिक्वेन्स जे AGCU आपण बघतो ते सिक्वेन्सेस आहेत. सो तो डीएनए mRNA मध्ये ती इन्फॉर्मेशन थ्रू mRNA ट्रान्सफर करतो, आणि मग जे mRNA ला जी इन्फॉर्मेशन तो पाठवतो ती त्याला बरोबर कॉम्प्लिमेंटरी असते. फॉर एक्झाम्पल ॲडेनाइन असेल तर आरएनए जो mRNA फॉर्म होतो, मेसेंजर आरएनए जो फॉर्म होतो, ॲडेनाइन असेल तर कोणता न्यूक्लियोटाइड असतो आरएनए मध्ये, युरॅसिल. ओके, तिकडे थायमाइन असेल तर इकडे काय ॲडेनाइन, ते आपण बघितलं. सो कॉम्प्लिमेंटरी असतो, म्हणजे त्या पेअर्स ठरलेल्या असतात, त्याच्यानुसारच तो सिक्वेन्स बनतो. सो डीएनएच्या इन्फॉर्मेशन नुसार तो सिक्वेन्स बनतो, आणि तो सिक्वेन्स मेसेंजर mRNA हा न्यूक्लियस कडून सायटोप्लाझम कडे कॅरी करतो, आणि सायटोप्लाझम मधल्या रायबोझोम या सेल ऑर्गनेल मध्ये तो ती इन्फॉर्मेशन, तो मेसेंजर आरएनए जातो. ओके, तो मेसेंजर आरएनए गेल्यानंतर आता पुढची प्रोसेस आहे ट्रान्सलेशन. आणि मग आपण कोडॉन आणि अँटीकोडॉन बघितले. सो मेसेंजर आरएनएची जी स्ट्रिप आपण बघितली त्यापैकी 3चा ग्रुप, एक 3 न्यूक्लिओटाइड्सचा ग्रुप, म्हणजे काय तर तो 3 ग्रुप हा एक अमिनो ऍसिड साठीचा कोड, ते 3 ग्रुप मधून 3 ग्रुप नुसार एक अमिनो ऍसिड बनेल. ओके, सो ते त्याला म्हणतात कोडॉन. आणि ट्रान्सफर आरएनए वर काय असतात तर अँटीकोडॉन, म्हणजे त्यालाच कॉम्प्लिमेंटरी असे न्यूक्लिओटाइड्स, त्याला म्हणतात अँटीकोडॉन. ओके, सो आता आपण या स्लाइडवर आलोय. ट्रान्सलेशन दॅट इज मेकिंग प्रोसेस प्रोटीन सॉरी. सो त्याच्यामध्ये काय होतं तर हा आरएनए मध्ये रायबोझोमल आरएनए मध्ये एमआरएनए आहे, ती इन्फॉर्मेशन घेऊन आलाय आणि टीआरएनए ते इन्फॉर्मेशनला बरोबर अँटीकोडॉन देऊन ते मॅच करते. जसं ते मॅच होतं तशा पद्धतीने त्या सेलमधून अमिनो ऍसिड्स अमिनो ऍसिड तिथे असतात, फूड थ्रू वगैरे जे काय तिथे बनलेले असतात, तिथे मिळालेले असतात, सेलमध्ये असतात, ते कॅरी केले जातात कुठे रायबोझोमकडे. आणि मग असा प्रत्येक अमिनो ऍसिड वेगवेगळे बनल्यानंतर त्यांचं सगळ्याचं बॉन्डिंग होतं, ते पेप्टाइड बॉन्ड्स, एक स्पेशल टाइप ऑफ बॉन्ड्स, त्याचं डिटेल नाही आहे आपल्याला. सो पेप्टाइड बॉन्ड्सने ते अमिनो ऍसिड्स बॉन्डेड होतात आणि त्याला म्हणतात, तेव्हा काय फॉर्म होतं तर प्रोटीन फॉर्म होतं. आता ट्रान्सलेशन म्हणजे काय, जसं आपण इंग्लिश टू मराठी ट्रान्सलेशन करतो तसं कोडच्या फॉर्म मध्ये आलं, डीएनए कडून वेगळं नाही का हे करत कोडच्या फॉर्म मध्ये आलं आणि ते कोडचं डीकोड झालं, म्हणजे आला कोड आणि त्यांच्यानुसार घेतलं अमिनो ऍसिड. सो हे झालं डीकोडिंग. कळलं, ते त्यांना कळलं. ओके, ते त्याचं त्यांना टीआरएनए ला कळलं, त्याच्यानुसार अमिनो ऍसिड आणणं. कळते, ते तसं आहे, नॅचरलीच ती गोष्ट तशी आहे, त्याचं त्यांना कळतं. सो समजलं. So that is nothing but translation. So let us see in English, decoding of mRNA into protein. एकदा mRNA च्या कोड नुसार प्रोटीन बनणे, अमिनो ऍसिड येणे ते एकत्र येणे प्रोटीन बनणे. सो त्या कोड नुसार प्रोटीन मिळाले, कोड नुसार कोड नाही जनरेट झाला, कोडच्या नुसार प्रोटीन मिळाले. Okay, so decoding of mRNA into protein is nothing but translation. So this process, the transfer RNA थ्रू जी प्रोसेस होते ती आहे ट्रान्सलेशन. आता तुम्ही म्हणाल ट्रान्सलोकेशन. सो मेन या दोन प्रोसेस आहेत, काय आहे ट्रान्सक्रिप्शन, ट्रान्सलेशन. ट्रान्सलोकेशन हा मधला पार्ट आहे, जो इथे आपल्याला या डायग्राम मध्ये सुद्धा दिसतोय. लुक ॲट द डेफिनेशन. ट्रान्सलोकेशन म्हणजे जेव्हा हे काम चालू असतं, कोणतं, जेव्हा ते मॅचिंगचं काम चालू असतं. सो प्रत्येक कोडॉन त्याला अँटीकोडॉन, कोडॉन त्याला अँटीकोडॉन. सो हे ऑब्झर्व करण्याचं काम हे रायबोझोम मध्ये चालू असतं. सो रायबोझोमल आरएनए, त्यातले आरएनए हे काम ऑब्झर्व करण्याचं काम करतात. दॅट इज नथिंग बट ट्रान्सलोकेशन. बघा, रायबोझोम कीप्स ऑन मूव्हिंग फ्रॉम वन एंड ऑफ mRNA टू अदर एंड बाय द डिस्टन्स ऑफ 1 ट्रिपलेट कोडॉन. बघा, ट्रान्सलेशन डायरेक्शन ही दाखवली, तो असं मूव्ह करेल, हे 3 झाले की पुढे मूव्ह करेल. सो दॅट इज नथिंग बट काय ट्रान्सलेशन, ट्रान्सलोकेशन. लोकेशन चेंज करेल ना त्याचं, म्हणून ट्रान्सलोकेशन. ओके, गेटिंग इट. सो ट्रान्सक्रिप्शन, ट्रान्सलोकेशन. So look at this animation. सो हा टीआरएनए येतोय, इथे अटॅच होतोय आणि त्यानंतर हे जे आहेत हे जे सगळे बॉन्डेड होतील, हे जे दिसतात हे जे सगळे हे बॉन्डेड होतात एकमेकाला अमिनो ऍसिड्स, आणि जे काय फॉर्म होतं ते असतं प्रोटीन. ओके, so now central dogma. So this whole process, protein synthesis, is central dogma. It is called as central dogma because it though it is performed outside the nucleus, but its information is hidden in the nucleus of the cell, that is at the center of the cell in DNA. Therefore it is central dogma. मध्यवर्ती प्रोसेस आहे ती, म्हणजे जो कंट्रोल आहे तिचा, तो त्या प्रोसेसचा कंट्रोल हा न्यूक्लियस मध्ये आहे, म्हणून त्याला म्हटलं जातं सेंट्रल डॉग्मा. ओके, सो आज आपण बघितलं, वी हॅव स्टडीड प्रोटीन सिंथेसिस इन डिटेल. एकदा मी रिवाइज करते. सो प्रोटीन सिंथेसिस म्हणजे प्रोडक्शन ऑफ प्रोटीन, मेकिंग प्रोटीन. प्रत्येक सेलला प्रोटीन बनवणं गरजेचं असतं, प्रत्येक सेल ही प्रोटीन बनवते आणि ते प्रोटीन बनवतानाची प्रोसेस, त्यामध्ये आहे ट्रान्सक्रिप्शन, ट्रान्सलेशन, ट्रान्सलोकेशन. सो फर्स्ट आहे ट्रान्सक्रिप्शन, ज्यामध्ये DNA स्वतः बाहेर येऊ शकत नाही, राजा आहे तो स्वतः बाहेर येत नाही, तो काम करून घेतो DNA. सो DNA टू mRNA, त्याची इन्फॉर्मेशन त्यांनी mRNA ला दिली. mRNA न्यूक्लियसच्या बाहेर येऊन रायबोझोम मध्ये गेला आणि mRNA नुसार, त्या कोडनुसार ट्रान्सफर RNA आले तिथे आणि ट्रान्सफर RNA कोड मॅच केला आणि त्याच्यानुसार ट्रान्सफर जो काही तो कोड म्हणतो की हा अमिनो ऍसिड घेऊन ये, तर त्याच्यानुसार तिथे अमिनो ऍसिड कलेक्ट झाले, ते अमिनो ऍसिड कनेक्ट झाले पेप्टाइड बॉन्डने आणि प्रोटीन हे तयार झालं. ओके, सो स्टडी दिस इन डिटेल. ओके, सो एक पॉइंट घेतला आपण, नेक्स्ट पॉइंट आपण नेक्स्ट लेक्चर मध्ये घेऊया. ओके, थँक्यू.